Today we're going to make a bracelet using this DNA codon table here. We have 4 bead colors, string, a permanent marker, fasteners, and scissors. Write your first name on the top line and come down to the table and find the letter. This codes for the amino acid isoleucine in this case, and the letters ATA. Write them up here on this line. Then go for the second letter, L, leucine, which codes for the letters CTC, write them down again. And finally A, which codes for alanine and GCT. Using these 4 letters that code for your name, adenine, thymine, cytosine, and guanine, we're going to take the colors that they represent and make a bracelet out of them, as they all code for a different bead color. We have 2 great examples here of what your bracelet should look like when they're complete. As you can see, no 2 look alike unless you have the same name. You can adjust the size if necessary. It looks like these bracelets were really meant to bead. Happy beading! DNA is composed of 4 nucleotides: adenine, which we've made pink, thymine, which we've made yellow, guanine, which we've represented as green, and cytosine, which is orange. We have 2 examples of DNA sequences here, and we've chosen the first one to use as our DNA template. Every nucleotide of DNA pairs up with another nucleotide to make what we call a base pair. Thymine always pairs with adenine, and cytosine always pairs with guanine. That's why we have T with A with T, C with G, and so forth. To make edible DNA, you'll need some licorice, colored marshmallows, toothpicks, safety pins, and tape. First, take 2 strands of licorice and line them up parallel to each other to make sure that they're approximately the same length. Next, start sticking the toothpicks into 1 strand of licorice, which represents the DNA backbone. Space out the toothpicks approximately 1 centimeter from each other from the top to the bottom. Take the yellow marshmallow, representing thymine, and slide it onto the toothpick. Match it up with its pink buddy, adenine. Continue matching nucleotides down the DNA strand, following the DNA template provided. So this is what yours should look like. Add the second DNA backbone to the other side of the toothpicks to complete the DNA double-stranded shape. As we can see here, the colors of the marshmallows perfectly match the colors of the nucleotides that are shown in the DNA sequence. Now we're going to twist this double-stranded DNA into a shape that's called a double helix, which is how DNA is commonly found in nature. This is what a double helix looks like. How cool is that? Now we're going to take our DNA strand along with some of the tape and safety pins, which we'll use to label the different parts of the DNA. Stick 1 safety pin into each different colored marshmallow and add 1 more to the DNA backbone. You may need to unpin it to stick it in. This is what mine looked like when it was finished. Next, we're going to take some tape and we're going to make some paper labels to stick onto each safety pin. So now you can see that we have labeled thymine, adenine, guanine, cytosine, and the precious DNA backbone. Beautiful, isn't it? There you have it, everyone. An edible model of what makes you unique, DNA. Hi everyone, today we're going to do a really cool experiment. We are going to extract DNA from strawberries. For this experiment you'll need a resealable plastic bag, some strawberries, a container with a coffee filter over it, some water, isopropanol, a mallet, measuring spoons, a toothpick, salt, and dish soap. First we're going to take some strawberries and put them in this bag and then crush them using this mallet. So first open up the bag. And throw a few strawberries in. I used 6 strawberries, but you can use a few more or less if you want to. Push out most of the air in the bag and make sure that it is very tightly sealed. You don't want strawberry juice flying out of the bag, as this will become very messy and you'll lose some of the strawberry DNA. Now for the fun part. Hammer the strawberries until they are crushed in a nice strawberry jam-like consistency. You don't want there to be big chunks of strawberries, as this gets in the way of draining the strawberry juice in the filtration step. Now we're going to take some soap and throw it into the bag, followed by salt and finally water. Take your measuring spoons and measure out exactly 1 tablespoon of soap. Be careful not to spill it anywhere. Unseal the bag, being careful not to spill the soap outside the bag. You may need to have someone hold the bag open for you while you pour the soap in. Now we'll measure out 1 teaspoon of salt. We're just going to use a regular iodized table salt here. Again, be careful not to spill it everywhere. Finally, add about 1/3 cup of water to the bag. Seal the bag tightly and roll it up to ensure that the liquid does not accidentally leak through the seal. Vigorously shake the contents of the bag to get a nice thorough mix. Take some scissors and cut off a corner of the bag, which we've tightly sealed with tape to keep the juice together, and pour it into the coffee filter to drain out the strawberry liquid. We're basically going to make a baker's icing dispenser here. Only cut a small opening, because if you have large chunks of strawberry left over, they may fall through the bag and rip the filter. Carefully squeeze the liquid out of the bag and try to avoid dispensing the foam if possible. As you may notice, the occasional chunk of strawberry jumped onto the coffee filter. Because of this, I took a spoon and scooped out the bigger strawberry chunks and threw them into another beaker. This prevents the coffee filter from breaking and allows the strawberry juice to drain through the filter faster and more smoothly. Basically, the science behind it all is complicated yet simple. The positively charged salt coats the negatively charged DNA backbone, protecting it from disintegration. The soap surrounds the lipid bilayers of the strawberry cells, which breaks the cells and allows the DNA to burst out. Finally, we're going to take some isopropanol, which is also known as rubbing alcohol, and carefully pour it into the strawberry filtrate. Pour it down the side of the beaker so that you create a separate layer. DNA is soluble in water, but it is not soluble in isopropanol and will therefore appear as a solid, something that we call precipitate. You can see a cloud-like substance forming in the liquid. This is deoxyribonucleic acid, or better known as DNA. So now you can see that our DNA has lifted up out of the isopropanol and it's all kind of stuck together. It's very hard to stretch apart because it's quite viscous at this point. You can play around with the DNA by swirling the beaker a bit. Notice that it stays together. Try to pick it up with a toothpick. You'll notice that it becomes one consistent white blob. If you try to pull it apart with your hands, you'll notice that it's very resistant. Isn't it awesome? Happy extracting, everyone! Thank you very much for watching!